In this video, you will learn how to read the results of the Kirby Bauer test. You remember that we made a bacterial lawn with dilute culture of bacteria over the surface of Mueller-Hinton agar, and then we applied antibiotic discs to the medium. Then the plate was incubated at 37 degrees Celsius for 18 to 24 hours. Since we started with a dilute culture, the bacteria will start growing at 37 degrees Celsius. As they grow, the antibiotics diffuse into the medium, creating a concentration gradient. Higher concentrations of the antibiotics close to the disc and lower concentrations further away. If the bacteria are susceptible to the antibiotic, they will not be able to grow in the presence of the antibiotic, creating an area of clearing around the disc called the inhibition zone. The further away from the disc, the lower the concentration of the antibiotic, the more likely the bacteria are to grow. The size of the inhibition zone is a measure of how susceptible the bacteria are to the antibiotic. Larger inhibition zones are created when the bacteria are susceptible to the antibiotic. Smaller inhibition zones are formed when the bacteria are resistant. Here's an example of a Kirby Bauer test after incubation. Some of the antibiotics are surrounded by obvious inhibition zones. Another has a small inhibition zone, and yet another lacks an inhibition zone. In order to standardize the Kirby Bauer test, the diameter of the inhibition zone is measured. In our laboratory, we use this ruler to measure the diameter in millimeters. The distance between these two small lines is 1 millimeter. A centimeter equals 10 millimeters. In this example, 5 antibiotics were used: ampicillin, chloramphenicol, kanamycin, penicillin, and streptomycin. Because the inhibition zones may not be perfectly circular, the diameter is measured 3 times, and the smallest of the 3 measurements is compared to a table of standards. In this example, the inhibition zone surrounding the disc with chloramphenicol is 28 millimeters.